Hey everyone, this is Krish Gonzalez, and it is week 6 of microbiology at LAPU. This week's lab video exercise is pretty unique. It's going to be pertaining to environmental influences on microbial growth. So we're going to be doing some salt tolerance testing and pH testing. So kind of what we're looking at, same sort of microbes we've been using throughout the whole semester. We've got our S. cerevisiae and then our S. epidermidis. We've got 3 sort of vials for each of them, so 6 tubes in total. And essentially they're going to have 3 different concentrations of NaCl. We've got a 1% NaCl, a 7% NaCl, and a 15% NaCl. So then we'll incubate our microbes and see what we get. So cool. Kind of overview of the whole lab. What we're looking at is, again, what sort of factors into microbial growth. So essentially we're kind of got 3 sort of broad areas that affect into that. You got temperature, oxygen availability, and pH influence, right? So temperature, obviously we know, is going to restrict things that grow, right? Enzymes will either break down or the microbe will stagnate, depending on how hot or cold it is. Oxygen availability will things are either anaerobic or aerobic. And then pH influence will just be, whether something thrives in a more acidic environment or a more basic or more neutral environment, right? Outside of that too, we'll also be talking on osmolarity. So that's essentially isotonic, hypotonic, or hypertonic environment, right? I'll start kind of with, top to bottom. Hypertonic, you've got all the water leaving the cell, so it's pretty much high concentration in the cell, low water concentration. Hypotonic, all the water is in the cell and the concentration has now left the cell. And then isotonic is kind of in the middle, right? Talking on those, right? So we've got, let's just say 2 classifications of microbes that deal with high salt concentrations, right? So you've got the halotolerant, right? So halotolerants can live in high salt concentration areas, but they don't need the high salt concentration to grow, right? They can live in there, but they don't need it to grow, right? Then you got the halophile. So the halophile actually needs the high salt concentration to grow, and it's going to thrive best in that sort of environment. So think of those really high, 20% salt concentrations. So cool. So now let's get started. We're going to hop into our lab. Let's get everything set up here. All right, we've got our flame going. Set. We're going to throw on the mask. And let's get started. So essentially, here's what we're going to be doing, kind of just to make sure we wrap up everything pretty quick, because it's kind of wash, rinse, repeat. So the salt tolerance testing, we're going to start with our S. cerevisiae, and essentially just go into that. So let's start by inoculating our 1% NaCl. So open this up, run it over the flame, inoculate it like we all know how to do. Stick it there. Let's inoculate our S. cerevisiae like we all know how to do. Stick that bad boy there. Cool. Alright, we got our pipette, and we're going to be taking 2 drops, and then we're going to be dropping that in there. So let's take 2 drops. Alright, we got 2 drops. And then let's take this, inoculate it here. 1, 2. Cool. And then let's this back. inoculate this, and then we're going to cap it. Cool. And then we'll continue the process for the rest. But that should conclude our lab video exercise. Thanks guys, have a good one.